last summer, so today I'll talk about some of the work that I've done as a postdoc and also some exciting preliminary results that we've generated so far at the PET Center. So today I'll focus on zirconium-89 labeled antibodies for PET imaging of breast cancer. So really the goal of my lab, I have two goals, which is to develop new imaging agents as PET agents to predict response to targeted therapy such as drug conjugates based on antibodies and peptides, and also look at new radiotherapy agents based on alpha particles. So today I'll talk about zirconium-89 in PET imaging, and an example of that using trastuzumab to detect the human epidermal growth factor receptor 2 in breast cancer patients, and also a new zirconium antibody for imaging a relatively new biomarker called glycoprotein non-metastatic melanoma B, or GPNMB, because there's an antibody drug conjugate that's currently being developed to target this new biomarker, and I'm hoping to predict response to this new antibody drug conjugate using the zirconium-labeled antibody. So briefly, I have no financial interest to disclose, although I'm collaborating with a company. Zirconium-89 is a fairly new radiometal. It is a transition metal, and it is here on the periodic table. It has a half-life of 3 days, which decays via 23% of the time by positron. This allows us to image it by PET, and it has a characteristic 909 keV gamma ray. It's produced in a biomedical cyclotron via the transmutation of yttrium-89 to zirconium-89. So the cyclotron then would bombard an yttrium-89 with protons. It would emit a neutron to create zirconium-89. So it's not a natural radioisotope. It doesn't have any known biological functions, and because the half-life is 3 days, it's relatively long, as compared to traditional radioisotopes, they can be used to image large biomolecules, such as antibodies, nanoparticles, and even cells. So you can track the trafficking of various cell types. So zirconium-89 pairs well with monoclonal antibodies because it matches the half-life of the biological half-life of the antibodies, which circulate in the body for several weeks. And because of this extended imaging time, we can allow the antibody to circulate in the body and clear, and wait for the tumor uptake to get better because we can reduce the noise from the rest of the body to allow us to give us increased tumor to background ratios. Some of the potential advantages of using zirconium antibodies for PET imaging is that you can assess the target expression of various receptors so that you can plan or adjust the treatments accordingly. So for example, if this patient has breast cancer, which may have a negative histological result by biopsy, it can have positive expression in a distant metastatic lesion, and then you can also see probably a heterogeneous uptake of the zirconium antibody. So this will really allow us to tailor which antibodies should be given to a patient for therapy. And finally, you can assess drug delivery. So antibodies are commonly used for therapy, and if we follow where it goes in the body, we can assess whether the perfusion in the tumor allows delivery of the antibody. Zirconium antibodies in general is a fairly new research area. In breast cancer, it began in 2010, and I'll discuss that in the next slide. And it's continuously increasing in the number of trials that are initiated. So these are the number of trials that are initiated on the clinicaltrials.gov website. Imaging antibodies in green, and zirconium and other tracers, I guess, in other cancers in orange. So this increasing trend with zirconium-89 is partly due to the availability of zirconium-89, as now it is commercially available, and several facilities are now having PET imaging scanners in the clinic. So the PET Center is one of those facilities that are capable of having this kind of trial. And that's why I'm here. So the other reason why zirconium antibodies are gaining popularity is because of the convenience and the ease of preparing the radiopharmaceutical. So zirconium needs to be trapped in a chelate. This is desferoxamine to complex it, to make it stable so it doesn't fall out and go to the bone in the body. And you would conjugate it via one-step reaction to lysine residues of the antibody, and then purify it, and then radiolabel it at neutral pH, and you'll get high specific activity, or the amount of radioactivity incorporated per mass of antibody. And then you can analyze and optimize this reaction to reduce the aggregation of the radiolabeled product and increase the yield. So an example of zirconium-labeled antibodies in clinical trial is to target HER2. We all know that HER2 is a very important target for breast cancer because of the antibodies that are developed to target and have shown clinical efficacy. HER2 is overexpressed in 20 to 30% of all breast cancers. We know that HER2 can change over the progression of the disease, and it has a very heterogeneous expression. Part of that is the mechanism of HER2 shedding, where HER2 extracellular domain can shed from the cell surface and making that tumor cell not effective for treatment with the antibody conjugate. So if we image HER2 in the body, we can assess the whole body expression of HER2, and it could predict response to the corresponding antibody therapies. Examples of these therapies are FDA-approved are pertuzumab and trastuzumab. They both bind to HER2 at different epitopes, and they have synergistic but different activities for inhibiting tumor growth. And also, trastuzumab was modified with a drug conjugate known as TDM1 so that it can deliver the drug specifically into the cell to kill the cancer cell. So this is the first in-human study of a zirconium-89 labeled trastuzumab in metastatic breast cancer patients that was conducted in the Netherlands. This is a very important study because it set the stage for the rest of the field. Because they were able to detect unsuspected lesions, importantly in the brain. And so the hypothesis here is that the blood-brain barrier is compromised in the metastatic setting that would allow the antibody to cross and detect this potentially positive for HER2 lesion. Also, this lesion in the spine is not, I guess they couldn't biopsy it, so they couldn't confirm HER2 positivity. But because there is uptake with the zirconium trastuzumab, we can see that, yes, there is a lesion there, and it could be HER2 positive. And so that might affect the way this patient is treated. So another important outcome of the study is that they were able to show that you need to inject non-radiolabeled antibody to allow or to prevent this first pass clearance. So if you only inject 10 milligrams of trastuzumab before you inject the zirconium antibody, most of the zirconium antibody would clear through the intestines. However, if you increase the dose to 50 milligrams, then you can see the zirconium circulate in the blood pool. And so this study paved the way towards other studies. And at Washington University, I was fortunate to learn from radiologists, breast oncologists. And as a radiochemist, I contributed the aspect of making the radiolabeled antibody. And I learned from radiation physicists who did the dosimetry studies. So I'll only talk about the first cohort of this study, which were 12 patients who were confirmed HER2 positive using biopsy. These patients were injected with 1 to 3 millicuries of zirconium trastuzumab, and the objective is to look at the dosimetry and assess the safety of the zirconium antibody. And so with this study, this is an example of one of the patients with metastatic disease. She has lesions that are presumed to be HER2 positive on the shoulder, on the hip, and on the thigh. The uptake is significant in the liver because that's where antibodies are metabolized. And so it's fairly difficult to assess hepatic lesions in the liver. but the optimal imaging time points is 4 to 6 days post-injection. There's about 20% injected dose in the liver, which is a dose-limiting organ, and overall the effective dose to the patient is about 17 millisieverts per millicurie. And what's important is that at these doses there were no adverse events. So another finding in this study was that there were differences in the number of lesions that were detected by conventional CT, so these are the number of lesions detected by CT, versus the number of lesions that were detected by zirconium trastuzumab, and that's mostly observed in the metastatic setting. So for example, in this patient who received 3 prior HER2-targeted therapies with metastatic disease, she had 1 lesion, and so the zirconium PET was able to detect that 1 lesion, plus detect 3 additional lesions. One of these lesions was later detected by CT 3 months later. So the power of PET really can help with identifying new lesions and also assess that it is HER2 positive. So then I thought, well, what happens to the triple-negative breast cancer population? If HER2 positive patients can benefit from trastuzumab therapy, what options do triple-negative patients have? And so I looked in the literature, and really, I really only found that chemotherapy is the only option for the triple-negative. And this study conducted by a group at Emory showed that at every stage of breast cancer, the triple-negative population has a poorer overall survival than the non-triple-negative breast cancer. So part of the reason for this gap is the improvement in therapies for the non-triple-negative breast cancer with the advances of targeted therapy. So I looked for potential new investigational targeted therapies, and I found Glembatumab vedotin, or CDX-011. It is currently in phase 2 clinical trial. It's the sponsor is Celldex Therapeutics, and the target for CDX-011 is GPNMB. So I read that GPNMB promotes metastasis and is associated with the risk of recurrence. And this antibody drug conjugate has this valine citrulline linker to an MMAE drug that when the antibody binds to GPNMB is internalized and goes to the lysosome where the enzyme cleaves the drug and prevents microtubule formation. So in this phase 1/2 clinical trial, CDX-011 was able to improve the overall survival of these patients compared to patients who were treated with investigator's choice chemotherapy. So I wanted to know whether PET of GPNMB using the naked antibody can select patients who might benefit from this antibody drug conjugate therapy. So to do that, first we needed to go to the basics, to the chemistry, and really characterize what the antibody is doing and how it's behaving. So when you modify the antibody with the chelate, there's a possibility that you can affect immunoreactivity. So we needed to check that. So by optimizing the conjugation reaction, we can show that the difference between the DFO conjugated versus the naked antibody is not very much, so we know that it retains immunoreactivity. I did some cellular internalization studies because that's the mechanism for delivery of the drug. It has to internalize into the cell. And at 37 degrees, 50% of the zirconium antibody is internalized within 30 to 60 minutes. I characterized the affinity, which is 25 nanomolar, and also with that, quantify the amount of GPNMB on the cell line that I was using, which is MDAMB 468. And so I also did some other studies to show that it really is specific to GPNMB expressing cell lines. So then I moved on to imaging of xenograft models. The 468 xenograft is positively expressing GPNMB as confirmed by immunohistochemistry. This is an immunofluorescent stain where the green is positive and the blue represents the nuclei, and 231 is GPNMB negative. Here's that tumor, and we don't see any expression on the tissue. So this is at 4 days post-injection. We know that at 7 days post-injection, this is when we quantify the amount of radioactivity that's taken up in the tumor that's normalized to the weight of the animal. The uptake of zirconium antibodies retained at 7 days. So this is the biodistribution study showing increased tumor uptake and retention in the tumor, and showing the metabolism or the clearance of the zirconium antibody from the blood and also from the liver. One disadvantage of the zirconium DFO complex is its propensity to go to bone over several days. So that's why you see in this gray line over here, we see increasing uptake of the zirconium to the bone. So at Yale, we were able to start the lab and do some animal studies. So here we tried to reproduce some of the work that I've done at WashU, and so did some inoculation of these cancer cells, let them grow to tumors, and then inject them with the zirconium antibody. And then at 7 days post-injection, we would image them using PET, and after that, then we'd inject with FDG, because that's the standard PET agent, and that's the FDA-approved agent to detect tumors. So 1 day later, we then injected the drug, the antibody-drug conjugate, and then tried to image with FDG 7 days after the treatment, because I wanted to see if there would be some correlation between the zirconium PET imaging versus the change in tumor metabolism by FDG. So these are some of our preliminary studies that my group has done a heck of a job doing and preparing. So this is the GPNMB positive tumor model showing high uptake. This is the standard uptake value here, and this is the negative, and this is the standard uptake value. This is the same mouse as this one. This one was injected with FDG following imaging of the zirconium antibody, and really doesn't show any tumor uptake. So we have to rethink whether FDG is worthwhile for us in the preclinical setting. This is the tumor growth curve after treatment of the antibody-drug conjugate. The spring-like colors represent the GPNMB negative tumor size, and the reduction in tumor growth is from the GPNMB positive tumor model. So then we tried to group them according to the response of the tumor, so shrinking of the tumor, greater than 50% reduction. We show that the GPNMB positive has high uptake with the zirconium antibody versus low uptake of the GPNMB negative. So this is still very preliminary, and we need to add more N numbers to our animal models. But there is a possibility that we can show that there would be a baseline or a threshold of positivity for zirconium CR011 that would correspond to positive GPNMB. So in future work, I would like to test this imaging studies in more clinically relevant models, the patient-derived xenografts, and also to translate the zirconium antibody to first-in-human trial. And so with that, I'd like to thank my former colleagues at Washington University. I learned so much from so many people with very different scientific backgrounds from me. And my postdoc mentor, who moved on to the University of Alabama at Birmingham, Susie Lappy, Celldex Therapeutics for providing the antibody and the antibody-drug conjugate. My team, who's done a fantastic job of learning new skills and also getting some data for you today. The PET Center and my mentors, and finally the R00 for funding. Thank you.
Are there any questions for Bernadette?
Why doesn't the FTG label the tumor? Did you?
Yeah.
They were both pre-injected with your agent as well. Did you say that? Did I
Yes.
get that correct?
Yes.
Did you ever inject any tumor alone? I mean, meaning a mouse that had never seen the first antibodies. Although negative should
Yeah.
have been affected by that anyway.
We still have to do that. There could be many reasons for why we didn't see any uptake in FDG. Triple negative is supposed to be avidly taking up glucose, so it could be logistics, and it could just be the tumor models that we're using are not.
They're not rapidly growing at that stage, perhaps, so they're not so glycolytic.
We have to do more more testing.
Have you ever looked at your HER2-directed approach to other cancers other than breast?
Not yet.
The reason I ask is that for some of the other indications that are HER2 overexpressing, it's clear that a probably more prominent mechanism of resistance is loss of HER2,
Yes.
particularly in gastric cancer, where that's the other indication that trastuzumab is approved. And it's been a challenge because when people want to then do subsequent trials in these populations, you have the archive tissue, which may be HER2 positive, but whether they maintain that in a dynamic sense is unclear, and that might be a great way for you to investigate the agent.
And gastric cancer is being pursued at MSKCC.
Yes, I know.
So okay.
So in the studies that you showed where there was a comparison of, I guess it was CAT scans or MRIs and the use of the labeled antibody, and there was a disagreement.
Uh-huh.
There were more tumors that were detected generally by scanning, by imaging, than with the antibody. Have those tumors been biopsied? Is there any evidence for why there's a difference? Is it loss of the antigen?
I
Or is
think-
it just a technical failure? Or?
Oh, that's still being investigated. That would be future work for the researchers at WashU. I think initially they couldn't get, or biopsies weren't part of the clinical protocol. They just wanted to look at the imaging quality and look at the dosimetry because that's the first step. And then correlating with pathology would be the next step.
Well, Bernadette, thank you. It's
Thank
great
you.
talk. So our second speaker, frankly, needs no introduction here at Yale, but I'll offer one anyway. So Richard Flavell is the Sterling Professor of Immunobiology and a member of our Cancer Immunology program for the Cancer Center. Richard is a fellow of the Royal Society, a member of the National Academy of Science, as well as the Institute of Medicine. And his lab, which has really been an innovator in immunobiology in terms of understanding adaptive immunity, T-cell tolerance, activation of the immune system, and building new models, has informed this field in so many unique ways. And among the many things he could probably talk about to us today, his topic over the next 25 minutes will be humanized mice for the study of human cancer. So Richard, thank you. I don't know what that is.
Updating PowerPoint, that's interesting, isn't it?
Oh,
I think I'm using the Galilean version of PowerPoint, actually. But, let's have a look here. Can we escape from this? Thank you. Just go away. Show details. Yeah, right. That's what we want at all. Let's get you out of the way. Well, and I've.
so you can close that.
Yeah, we'll close you, and then I'm going to be under here somewhere. That's it. Yeah. No, thank you. All right, let's give this a try. All right. So it's a delight to be here. I'll talk to you very kind of briefly about our work in the area of humanized mouse, and the idea, of course, is to model cancer and hopefully do something useful for patients one way or the other. The humanized mouse is an old idea. It was developed first by a whole lot of people back with the Hugh SCID models and this, that, and the other. The main problem has been that the earlier versions, and still today, there's a lot of things that need to be improved. We focus on the hematopoietic immune system. You'll see why in a short while. And if you look at the comparison of real people to the various mouse models, you can see there's a lot of deficiency in all of these models which have been developed. The most well-known, of course, is NSG mouse, the nod-skid gamma common mouse, but it, like all the others, suffer from a large number of deficiencies. And the particular interest that we have had over the years, and I'm sure you all, is the cellular composition of the tumor microenvironment and the role that these cells play in the growth and immune response to the tumor. Particularly, of course, interesting now, immuno-oncology has become such a vibrant field. And one of the things that we wanted to focus on, has actually been myeloid cells. Myeloid cells in tumors, of course, famous for influencing tumor growth in a variety of important ways. And so what are humanized mice? Well, the way we do this, it was based upon what Marquez-Nostra developed in a paper in Science in 2004, which is to use human HSC populations using CD34 cells, inject them into newborn mice, into the liver. Those cells will engraft nicely in the liver because the liver is a hematopoietic site. And but when the mouse grows up 8 to 12 weeks later, you can get a reasonable development of the immune system with the deficiencies I'll get into in just a second. What did we do that was different? Well, we noticed that there were many populations that were very poorly represented. And the idea that I had right back then, this was in 2005, was that this would be a problem of the cytokine microenvironment. And that makes sense because a lot of mouse cytokine genes are very poorly conserved if you compare them to humans. So it therefore made sense that we knew they didn't work to grow human cells in vitro, they wouldn't work to grow human cells in vivo either. And so if you look at all the bad ones, the red ones and the yellow ones, there's plenty of these things that are critical for hematopoiesis that literally didn't work. So what we did is to team up with Regeneron Pharmaceuticals, who had developed a quite high throughput way of making gene substitutions, and we took the mouse and inserted the human gene for a given cytokine into the mouse locus. And what that we hope would do, and it actually did, is to make the cytokine at physiologic concentrations in the right amount, the right place at the right time. And it basically does that. And these are some papers that summarize the concepts and outcomes. Fast forwarding to maybe 4 years ago, we had made mice with these individual cytokine combinations, and eventually we put these together by breeding into this mouse, which one of my postdocs had the great idea to call Mr. G, which is a catchy name, which we still use. And the purpose of these genes are first of all, RAG and gamma. This is like an NSG mice. This is for immunosuppression, so you don't get mouse TB and NK cells. SERP alpha is an important molecule in macrophage tolerance. This combination, SERP alpha and CD47, is actually very interesting in cancer therapy concept these days. This prevents the mouse macrophage eating human cells, and this is actually a problem in humanization. But the problem is that mouse SERP alpha doesn't really bind well to human CD47, except in a few cases. And so what we did here is to substitute the human SERP alpha for the mouse, and that actually makes this work much better. So this provides us with phagocytic tolerance. And then these two molecules, or three molecules, provide the development of myeloid cells, which doesn't happen in the absence of these molecules. We then, in early studies, Anthony Renveau in the lab compared these four groups of mice. So these are these mice with or without SERP alpha, and this is NSG mouse, the commercial one, and this is RAG gamma, the version with the BALB/C background. And you can see that there's significant differences in engraftment. If you focus on the far side here, each dot is a mouse, so Anthony did vast numbers of mice together with Tim Willinger. And you can see here that RAG gamma is quite poor, NSG is better, but these strains actually are better still. And the main reason that they're better is they give you much better engraftment of myeloid cells. So this is NSG compared to this Mr. G strain, you can see that in blood, for example, there's a lot more myeloid cells, and this shows the composition of blood in those mice. I'm going over this quickly because it was published a few years ago now. Myeloid cells in red. It's still not as good as human. The reason for that is mainly because we don't get very good granulocytes, and we're still working on that. We get tissue macrophages, which is very helpful and important. So you can say NSGs, of course, don't do this because they don't have the myeloid ingredients. They don't have that MCSF or CSF1, but the Mr. G mice have human cells in the lung. They have human alveolar macrophages. This is all, of course, post-engraftment with human CD34 cells. So these are alveolar macrophages. Liver has macrophages, including Kupffer cells, we believe. The colon also has human macrophages. And in blood, there are the three sets of monocytes, which you find actually in people. And we've gone on to characterize those. I'll skip over that today because the talk is quite a short one. But the bottom line is that, again, this is an improvement over the NSG system, both quantitatively, which you can't really see from a FACS plot, and qualitatively as well, having these additional populations. The innate immune responses of these mice, as a result, are much better. Here you can see this is infecting a mouse with listeria, and you can see NSGs don't make much of a human cytokine response, but Mr. G mice do very nicely with that. You can see here, this is looking at IL-6. It shows interferon responses in the lungs of these mice. This is a result of those alveolar macrophages. Although, of course, remember, we don't have human epithelial cells, so we lack that component in the flu infection as well. We collaborated with Stephanie Helene's lab over the years, and Stephanie and our colleagues showed, in fact, that something which we were interested in, which is what is the origin, what is the cellular origin in the bone marrow of the human cytokines. And you might not be surprised by the fact that this actually is a fibroblastoid stromal cells, and you can see in the Mr. G mouse, you can detect GM-CSF, MCSF, thrombopoietin, all of the cytokines we had been looking for, with the exception of IL-3, which is made by T cells, actually. So to summarize that, this was already a good improvement over the previous generations with regard to myeloid cells, and gave a mouse which looked potentially useful. But what about functional utilization? Well, one interest that we have is to study human NK cells and human CD8 T cells. And we hypothesized, actually, that myeloid cells were known to be a source of a second cytokine, interleukin-15. This is required for NK cells and for CD8 T cell maintenance. And so the idea was that the human myeloid cells might make the IL-15 in the right environment and therefore allow these other cells to be produced. And that turns out to be correct. This is the way this works. I can skip it. And this shows that indeed, if you looked at the Mr. G mice, they made IL-15 mRNA and they made IL-15 receptor. This is made by in the same way. And particularly, it was this class of monocytes which did it. And if we looked in the blood and various tissues of these mice, very reassuringly, this is NK. These are NK cells in various places. Of course, the place you find a lot of NK cells in the liver, and the lower panels, you can see it's the Mr. G mouse, have a lot of NK cells in the liver. The NSG mice don't, because they don't make the IL-15, because they don't have the myeloid cells. And so this kind of gives you an idea of the quantitation. Each of these, again, is a mouse. So this worked very nicely and allows one to study NK cells. There, in fact, are killer cells. They will kill in vivo if you put a tumor cell in there. More about this in a minute. And they will degranulate and make interferon gamma. That's what was being observed over here. So, again, these are functional NK cells in these mice. So that, to summarize the first part of the published work, really, the Mr. G mouse, by substituting cytokine genes, or human cytokine genes, does allow this production of these molecules, which allows myeloid cells and, in turn, other cells which are dependent upon them. More recently, we've utilized a mouse by knocking in human IL-15, and that may seem contradictory for you, but actually IL-15 is a stromal cytokine as well as an immune cell cytokine. And so Dietmar Brandstetter and Liang Shan collaborated with Kagan Gura from Regeneron. Kagan and his colleagues made the knock-in IL-15 mouse. We combined that in breeding with the SERP alpha. Remember, that's the phagocytic tolerance and the rad gamma, and made these SRG15 mice. And these two guys studied all the biology of these mice. This is we now by this time a knock-in of SERP alpha. This works nicely to engraft, and I'll skip that. Just take a word for that. And then we knocked in also human interleukin-15 using these methodologies, velocigene stuff from Regeneron, and very nice expression of IL-15 through a stromal environment in liver, lung, small intestine, which is where we're expecting this. And sure enough, this molecule is functional. So one of the things about the intestine is there are these unique intraepithelial lymphocytes, which are also IL-15 dependent. And these cells actually develop in the small intestine of these mice. And you can see these cells are being detected here by, you can see there's an order of magnitude more cells compared to the mouse which lacks IL-15. You can detect them by staining. This is where the red arrows are. The cells in the SRG mouse, you only see cells in the lamina propria. That's the immune organ underneath the epithelium. But in the epithelium itself, you only see these cells in the SRG15 animals. They have special properties. I'll skip that because it's not a particularly cancer-oriented topic. But relative to the cancer story is the fact that these mice support good human NK cell development. Same as I basically told you a short while ago. The effect of IL-15 is on the mature NK cells, not the precursors, as we would have expected. And if you look again in the comparing NSG to the SRG15, if you look at, for example, liver here, you have many, many more NK cells than in the NSG equivalent situation. But now we're supporting them through the stromal production of this molecule. This is all the counting of those cell types. These cells are cytotoxic. The mature NK cells in humans are thought to be CD56 dim, etc., etc. And these cells are nicely cytotoxic. They will kill human cells. I'll skip over the details of this. We've done cytoff and characterize these cells, and they basically look pretty like human NK cells with a few differences. And here again, this is doing that overall comparison of human to this cell type. Now, these cells are also functional. If you transfer immune cells, you transfer MHC knockout tumor cells, they will kill them in vivo. This is the number of cells left. This is after they were injected into the 15 mouse. This is what we see there. And finally, for therapeutic antibodies like Rituximab, that's the anti-CD20 therapeutic antibody used for enormous numbers of indications. This is an antibody that actually requires NK cells for its function, and the reason is that it requires that is that acts by killing, for example, lymphoma by ADCC, antibody-dependent cellular cytotoxicity. And so we wanted to see whether we could model that in this SRG15 animal. And that turns out to work. We used Rituximab to treat Raji cell. Of course, this is a cell line at this stage. Putting this into mice, and you can see the mice labeled here. They're then treated with Rituximab by that protocol. And you can see that the Rituximab, if it goes into the SRG15 mouse here, clears that tumor, but it doesn't work if you put it into an NSG mouse. Here's an NSG with the antibody. This is SRG with the antibody. Only works if you have the IL-15 to provide the NK cells. So this is the weight of the tumors. This shows, again, basically a confirmation of that, and using a non-engrafted SRG15 as an additional control. So this doesn't have the human cells. This one does. Both have the reagent. So it is dependent on the human engraftment to work. So to summarize that, it looks like this model of NK biology is useful, and it allows us to study certain aspects of at least tumor therapy. And then finally, for the purpose of today, I'll talk about our steps towards making patient-derived humanized mice to study anti-tumor immune responses. And of course, this is perhaps the most interesting area in general because of the potential for immunotherapy of so many of these diseases. Now, in the first experiments, we took the Mr. G mouse, the one I started with earlier in the talk, we engrafted and then we implanted human melanoma cell lines. These are recently developed human melanoma cell lines. And then we studied the interaction of the immune cells. This all in collaboration with Carolina Paluca up at the JAX and Jan Martinek from her lab. They were at Baylor at the time, they're now up at the JAX. And then, so these, we did this comparison with NSG versus Mr. G again. As you know, the biology of macrophages is quite interesting, and tumor macrophages are, of course, of interest because they can promote tumorigenesis and metastasis. So there's a strong correlation, actually, between macrophage density and poor prognosis in people. And so we did staining of these tumors, and maybe it's easier to just focus on the numbers here. So NSG mice, of course, you don't have any infiltrating macrophages in the tumor. And remember, these mice have received human stem cells providing the immune system. They receive the tumor cell line as well. And you can see NSG doesn't populate the tumor with human macrophages because it doesn't have very many. The Mr. G does that very nicely. Patients have more sometimes. This may be something to do with neutrophils. Not sure, but either way, it seems to work quite well. And you all are very well aware of the M2, M1 cliche for the separation of different kinds of macrophages. In particular, tumors are thought to carry these M2-like macrophages, which are producers of growth factors and things like VEGF, which of course can mediate angiogenesis. So we got pretty interested in that. This is just staining to show that CD206, which is an M2 marker, can be detected in both Mr. G tumors as well as real patient samples. So we went looking for angiogenesis because of VEGF, which promotes angiogenesis. The hypothesis was that the human myeloid cells would be making the VEGF, which would help the angiogenesis. So we studied that. If you stain for human endothelial cells, the Mr. G tumors are much better vascularized than the NSG equivalent. And that affects the growth, actually. Now, this is looking at NSG mice and Mr. G mice which have no human immune cells, okay? And then this is the tumor we grow after whatever the time point was. You can see there's not very much difference between the tumors. But if you put the same tumors into engrafted mice that have human immune cells, then you can see that the human tumors are bigger and they look better vascularized by obviously a very low resolution technology there. But here you can see that this is unengrafted, so this is very statistically significant. This is NSG engrafted, and this is Mr. G engrafted. So there's a big increase in tumor volume, and all of that seems to be dependent on vascularization because we treat those animals, these two groups of animals, with Avastin, obviously blocking the VEGF system. You can bring them right down to the unengrafted mouse. So that was an interesting study with that particular animal. So again, this means that this model could be useful for further drug testing. The other interesting thing is that in further studies we've noted that actually human hematopoietic cells support human tumor metastasis. This is with the same cell line that what we were just talking about. This is a non-engrafted Mr. G with a sub-Q melanoma cell line. And this is looking at engrafted Mr. G. You can see that in this case, these metastases are very easily detectable and shown by histology to be the correct thing. So that's of course, confirms a lot of what is known in the mouse systems, but it does allow a study of this in human, and that Anthony is working very hard on, as well as Michael Curazzi, who's in our department. So where are we going? Well, we want to go to a more patient-oriented study, and so what we're heading for is to have patient-derived hematopoietic cells and primary tumor from the same patient. And so Michael, who's been working on this, has been studying this in both lung cancer and in collaborative studies on melanoma. And I'll tell you a little bit about that in the amount of time that I have left. And so what we first took was two patients who had tumor and blood cells harvested back actually in the early 2000s by Carolina's group, and this shows engraftment of these humanized Mr. G mice, looking at the various cell types. You can see myeloid cells in red, and what this shows is that we could detect from the patient samples NK cells, T cells, B cells, and myeloid cells in some of the cases. And so we went to look and see what effect this engraftment had on the growth of the tumors. And you can see that in the next slide. This is a very early experiment. And so what you can see here is the non-engrafted mice here. It's a small number of animals because there were small amounts of material available. So we put 100,000 tumor cells in there and looked at after 6 weeks. You can see this is this tumor. In this case, there was no increase by the adding the human immune cells, but in this case, the tumor grew better and generally, and this has turned out to be reproducible in these cases. And what we're finding by looking at more and more samples, maybe I'll have a couple more going into this, is that we can recapitulate this additive effect of having the human immune cells on the engraftment of the human tumor, even when in the case that the cells are derived from the same patient. So Michael has been working on this now for quite some time, developed a very nice system, and these are his collaborators in the lab, Yun Jiang, who's done some work developing a new mouse system. I'll tell you briefly about this one. This mouse system is which is the state of the art, the one we're using now, which is we've added a single component, a knock-in of interleukin-6. The reason we do that is because this increases the engraftment. Turns out to be very important because when we're taking patient samples, we're almost always using human adult bone marrow, and the engraftment in mice is much better with fetal or cord blood material than it is with adult. So we need all the efficiency we can get, and this IL-6 helps us a lot with that. So this is the mouse, Mr. G6. It's the same as Mr. G, except it has the IL-6. The white blood composition is very similar, and you can see here that this is just looking at one example, comparing Mr. G, which was obviously I showed you earlier was good, but here you can see, and this is Yun Jiang's data, that Mr. G6 is demonstrably better than that. And she's done many experiments of this nature using adult CD34 cells. This really explains the difficulties that we've had with achieving good engraftment in the adult system. And what I'll try and show you, this is now looking at fetal cell engraftment, again to give you an idea of efficiency. This is looking at blood immune cells, NSG, Mr. G, and Mr. G6, which again shows quite some improvement using 100,000 cells. And it's very sensitive to cell number. This is comparing Mr. G6 using, again, fetal cells, but you can see here, you can get a pretty good engraftment by only injecting a few thousand CD34 cells, which is a very small number of real stem cells, presumably. So it's a very sensitive system. But again, the problem, of course, is dealing with adults. And we can skip that, I think. With adult, you have to add very many more cells, and we're still working at getting that optimized. But Michael has been able to get significant engraftment now with patient material, which is a great relief and stimulus for the studies. Let's keep that as well. So Michael has been collecting non-small cell lung cancer samples and autologous bone marrow aspirates. I'm sure he's spoken about those at various talks at the Cancer Center. And we're now working with these. This is the whole protocol that he's working with. And I'll just show you a couple of samples. Maybe I'll skip ahead and find you one of the nice growth curves he produced. So here, for example, is looking at engrafted versus unengrafted mice for the autologous humanized mice that he's generated. These are melanoma samples, but the principles are similar. The non-engrafted animals, meaning non-engrafted, meaning not having the immune cells, being in general growing more poorly compared to the engrafted cells. So that it looks like he's well on track to study these very interesting problems, and by combining all the modern technologies of single-cell RNA sequencing, engraftment of these animals, and CRISPR mutagenesis, I think there's a lot of really exciting things that he's planning to do with this. Time's pretty much up. So what's next? Well, lots of stuff. He's now studying these systems in a whole variety of different ways, as I explained. So I'll stop there. Thanks very much for listening to me, and sorry if I went a little quickly, but hopefully it was not too hard to follow. This is the current crew. Mike, I've just mentioned already, Yun Jiang, who's been working very hard on getting both the Mr. G6 system working and also getting granulocytes working. Liang Shan just left our group. We returned Wash Yu the favor, and now we sent Liang. He's now working down there actually, and so with the immunology group, and he's continuing this work. And Anthony Renvaux, who was really a pioneer in our lab, now has a job at the Fred Hutchinson Cancer Center, so we're populating some very respectable places around the country, and we're continuing to do this work. I mentioned Jan and Carolina, the Jacks. Carolina, of course, are very well-established scientists, and these are other people that have worked with the project. Thanks very much.
Thank you. Questions?